Last week, we looked at mix and match vaccines. Though preliminary studies on this strategy have been encouraging, some experts have raised concerns over combining shots without proper evidence. And what's the rationale behind this warning? We break down this issue and more in the next couple of minutes. I'm Nidhi Kumar and you're watching Science Time, a show that brings to you the best that science offers, from exciting developments in science and technology to futuristic solutions. And let's move on to story number 1. The mix and match strategy has generated a lot of interest in the recent past. It is touted to address the problem of vaccine shortages. A preliminary study suggested that one dose of the Oxford vaccine sold in India as Covishield, followed by a Pfizer booster, generated a robust immune response. It might also offer better protection against new variants. The Russian Direct Investment Fund has tested the combination of Sputnik V and AstraZeneca. The results of their study are expected to come out in sometime late July. In a nutshell, we have only evidence from preliminary studies as of now. Though encouraging, the jury is still out on whether mix and match vaccines are safe and effective. Studies evaluating the safety and the immunogenicity, which is the ability to evoke an immune response, are still awaited. We might gain a better understanding in the days to come. And in the meantime, get yourself vaccinated as recommended to lower your risk of contracting COVID-19. And with this, let's move on to story number 2. And the pandemic introduced us to new vaccine technologies, from RNA to DNA vaccines to protein-based ones. Now scientists are studying new technology, and it is already showing some promise. According to phase 1 clinical trials, the technology is called self-amplifying RNA, or saRNA, and is related to the messenger or mRNA based vaccines. mRNA vaccines carry a molecule that contains instructions to produce a part of the coronavirus spike protein. Once injected, our bodies read these instructions and make the spike protein. By introducing a part instead of the whole of the virus, mRNA vaccines teach our immune system to fight SARS-CoV-2. The saRNA also follows the same principle. In addition to carrying mRNA to the body, it will also ferry polymerase, an enzyme that makes a copy of the RNA molecule. As a result, the body will exponentially produce mRNA molecules, making more of the spike protein. In theory, the saRNA shots could generate a better immune response with fewer side effects at lower doses. And to test this, researchers from Imperial College initiated phase 1 trials in 192 volunteers. Phase 1 trials are designed to test safety and immunogenicity, which is the ability to trigger an immune response. Their results showed that about 87% of the participants produced antibodies against COVID-19 following vaccination. This suggests that saRNA is immunogenic. Further, the researchers say that side effects were fewer in numbers, establishing safety. The findings are a part of the preprint study that is yet to be tested for accuracy. According to the team, the next step is to test for the efficacy of the saRNA vaccine, and the researchers are also working on a second version of the vaccine that generates a more robust immune response. Moving on to story number 3. Well, fish are the new meth addicts. Sounds astonishing, right? Well, it is true. A study from the Czech Republic has found that freshwater fish are becoming addicted to methamphetamine due to the accumulation of the illegal drug in water bodies. We humans have ourselves to blame for this. Many of the drugs that humans consume end up in water bodies. There are no efficient ways of treating this waste. Research has shown that drugs such as fluoxetine, also known as Prozac in water can embolden fish and alter their behavior. The latest study on this, published in the Journal of Experimental Biology, shows that illegal drugs like methamphetamine are also accumulating in the waterways apart from prescribed medication. To study this, the researchers decided to investigate whether brown trout fish are at risk of getting addicted to illegal methamphetamine. The brown trout were isolated in a tank of water laced with methamphetamine for 8 weeks. The water contained a concentration of 1 microgram meth for every 1000 liters, a level that has been found in freshwater rivers. And the team then transferred the fish to a freshwater tank and offered them a choice between freshwater or water containing methamphetamine every alternate day for 10 days. If the fish were addicted, they would feel the effects of withdrawal and seek the drug when available, the team said. Soon, it became clear that the trout that had spent 8 weeks in the methamphetamine contaminated water had become addicted to it. The fish in question suffered withdrawal in the first 4 days after moving to freshwater. The addicted fish were less active than their unexposed counterparts, the researchers explained. And what's more, the team found evidence of the drug in the fish's brains up to 10 days after the methamphetamine was withdrawn. The researchers said that the drug addiction could drive fish to congregate near unhealthy water treatment discharges in search of a fix and disturb their natural tempo of life. And such evidence of drug addiction in wild fish is another example of the unexpected pressure on species living in urban environments. I hope the study forces us to find ways to treat or better dispose of our own waste. And with that, friends, this is a wrap on this edition of Science Time. Stay with us every Friday at 9 p.m. Keep watching India Science. Namaskar.